Thank you very much for the invitation and for the introduction. Good afternoon, everyone. I'm very happy to be here and talk about something that I'm very passionate about. What I want to do first is give a very short global introduction to what is a digital twin, and then I will, speed dating-wise, go through some of the applications that we're working on in the lab. I will not go into depth in any of them. It's more of a focusing on the what and the why. If you have questions, please don't hesitate. I'm happy to talk at length about any of them, but I thought it was more interesting to show you a little bit of the diversity of what can be done in terms of digital twins. And then at the very end, I will use a few minutes to indicate where the European Commission is going with this. The VPH, the virtual physiological human, that was indeed started quite some time ago. Now they're going towards the virtual human twin, and so I want to inform you what this is about. Good. Let's start with first the concept of digital twins. Geert already indicated that this is something that is coming from the engineering field, where they have a very strict definition that says a digital twin is a virtual representation of a physical object or a process that is in direct interaction with that object or that process. So that means that there is a real-time readout of the information from the object or the process, and then a way of dynamically interacting with that process if the predictions indicate that, for instance, the process is going in the wrong direction. Now, imagine applying this in a healthcare or a biomedical situation. What does that look like? How often do we have enough readouts that we can make a model and keep it updated in real time? I think there are very few cases in clinics or in science where that is the case. Dynamically recalibrating an organ, that is even further from what we can easily do. So when we're talking about digital twins in healthcare, we went for a bit more of a global definition that says that it is the use of personal specific modeling and simulation in all areas of health and care. So from prediction, prevention, prediction, diagnosis, therapy, selection, and follow-up, but also not only of the patient or the process, but also looking at the design, the development, the optimization, de-risking of therapeutic strategies. So is there then no interaction? Well, for some people, they're very passionate about this need of having the link between the digital and the physical in order to call something a twin. So yes, of course there is, because we make a model that is using some information of the patient. It can be imaging data, it can be some blood data, in order to inform the model to make it personal. And then there is a human-in-the-loop decision-making. Somebody is making a decision using some input from this digital twin. So rather than having a semi-automated automated interaction, we have a human-in-the-loop there. Now, digital twins, I will be showing you mostly models. Digital twins, of course, they need data. They need sensor inputs, they need, this can be imaging input, and then, of course, we also need hardware to do the compute. We have the middleware to make the connection, and then we have the software that is looking at the models. Now, digital twins cannot be, or if we talk about digital twins of a person, this is not one huge model that we are working on. Digital twins are there to answer questions, which means that we try to put things into the model that are necessary, and not more than that, because that will also complicate the regulatory process if we then want to get it used in some context. So digital twins can be digital twins of the cells, as you will hear later as well, but also tissues, organs, or the entire system for specific questions. They can also be digital twins of devices, and what you see here is the left atrium mirrored of the heart, where there is an appendix that sometimes is responsible for the formation of blood clots. And so the gray thing that you see is actually an occluder device that needs to be implanted in order to shield that or occlude the appendix so that blood cannot get in there. And the planning of the surgery of this occluder device, the choosing of the right shape of the occluder device, that is already being done using a digital twin of the geometry of the patient. So this is something that is already in clinics. You can also have digital twins of processes. If we are using bioreactors to culture cells in 3 dimensions, we do not know very well, or we cannot measure at the level of a cell what is the environment that they're experiencing. So there you can use digital twins of the process that you can feed with sensor information from the entire culture to inform the twin. And then finally, you can also have digital twins of entire facilities, like we had during COVID, to manage the patient streams. but in general, this is used to manage the logistic streams in larger organizations. Now, when I say a model, what does that mean? What kind of modeling technology do we use? For some people, everything is AI. Now, there's actually a spectrum of in silico technologies. And if we put it on from left to right, going from black box, meaning purely data-driven, no mechanisms involved, to on the right-hand side, white box, which is purely physics-driven, so we know everything that is going on, you can put all of the models on a spectrum. You have AI models, especially when we're talking about image analysis. AI has been shown to have good results there. Actually, everybody is using some kind of white box model, because if we want to transition from the raw data from an image to the image that we can analyze, you're using physics. So you're actually using a white box models. But for most other cases, the in silico models are somewhere in between. We can use some mechanism that we can complement with data, or we can use some data, some AI that we can complement with mechanism to avoid hallucinations. So how can we apply this to the skeleton? How are we doing this in the lab? And these are the wonderful people that are showing the work, that are doing the work that I get to show today. So I have well, the first group of models are models that have to do with anything that happens inside the cell. So we're working on models. We have a skeletal cell atlas, a regulatory model, and we can do network inference. Now, just to go a little bit more into detail, with a skeletal cell atlas, everybody knows the bioinformatics, you can develop these atlases, and how are we using them in the lab? Well, as a blueprint, because a few days ago, one of my colleagues, Professor Papantonio, showed the work that we're doing on organoids in the cell, in the lab. So we are taking organoids of skeletal progenitors, pushing them through chondrogenic differentiation, and then we're trying to recapitulate the developmental process. We can do single cell sequencing, and then we're now working on the mapping of these individual fingerprints on the skeletal cell atlas in order to see how far we are from where we actually wanted to be with our cell culture process. Now, this is like using or building a static map. Like an atlas is like a paper map. When you're not at the right place, it doesn't tell you how to get there. It's not Google Maps, right? Telling you exactly what route to follow. And this is why you can use models to develop an executable chondrocyte. Here we worked on a model that had a protein and a gene layer with the main pathways that are involved in chondrogenic differentiation. We first did it on the growth plate and then extended it also to more chondrocyte or articular phenotypes. This was first done in a knowledge-driven manner, so PhD student going to the literature, making all of these annotations saying where every arrow in this network was coming from. Of course, you can also do it in the other way around. You start from all the data sets, you aggregate them, making sure that you're taking out all the batch effects, and then taking this integrated data set and extract from that the links between genes that we might have forgotten in the original network. Now, using this, we can then look at whether this network, if we make it a dynamic model, is capable of capturing the main states of a chondrocyte, which we were able to do. And then you can use this model to see, okay, if my cell is in the wrong state, if it is on a pre-hypertrophic path, and I want it to stay stable, I want it to stay a healthy chondrocyte, how can I do that? You can interrogate the model, and you can ask it, what do I need to work on in order to get my cells from blue, which is going towards hypertrophy, back to the orange situation that you see on the graph. So the model can make some suggestions, which we then tested in vitro in ATDC5 cells, but also in donor cells, where we indeed see that what the model predicted, we also see in the cells. And of course, this was just one factor, a very generic factor. You can do that with multiple factors, where you see left the in silico predictions and right the in vitro validation. This is one intracellular model. We can then use this also to integrate this with things that happen at different length scales. So what you see here is a model, a multi-scale model of osteoarthritis, where we take a model of the gait, or a model of the knee joint that is fed with information from gait, where we calculate stresses and strains in the articular cartilage, which is then translated into activation of the integrins, which is translated into an activation of the intracellular module. So this is, of course, It includes many simplifications, but the way these models can be used is to ask questions and to look at mechanisms and see if we integrate information, whether we get to something that makes sense. Another series of experiments is, for instance, devices, looking at microfluidics, looking at biomaterials, looking at bioprinting, and looking at bioreactors. If I focus for a bit on the biomaterial optimization, what we did there is working on bone scaffolds or bone tissue engineering. If you look at the literature, there are a lot of, especially also calcium phosphate-based biomaterials, same now with the hydrogels that are coming up, with a lot of different factors that are influencing the process. Now, when we were studying in the lab cell growth and bioreactor studies in titanium 3D scaffolds, you see these kind of curvature-based growth. So cells produce neotissue and grow faster in the corners of the unit cells. This is something that was not something that we observed for the first time. There's actually a whole field called curvature biology that focuses on this principle. But the nice thing about it is that you can translate this into mathematics really easily by focusing on this interface, the red line that you see on the right, the interface between what is neotissue in green and what is void. The moving of this red line, you can simulate it very easily based on effects that are coming from the local curvature, but also you can add effects of oxygen, nutrients, flow, etc. Just focusing on the curvature, this is again on titanium scaffolds. We tested different unit cells. So you take the scaffolds that you see on the right. These are 3D scaffolds with different unit cell shapes. We see them with cells. These are periosteal-derived cells that we cultured for 7 and 14 days and then quantified the amount of neotissue that was formed. On the left-hand side, you see the predictions of the graph. We used the green point at 7 as a calibration point to see what is the correspondence between compute time and physical time. And then everything else actually uses that only calibration point. And you see that both quantitatively in the graph and qualitatively, if you compare the two middle columns here, we get a good correspondence because the curvature-based growth is something that has been shown over and over again. We then did another experiment to show that also in calcium phosphate-based scaffolds, where there is more activation of the cells, we still had that same phenomenon of curvature-based growth, and then used this model to look at 3D scaffolds that had more interesting shapes for the cells. So we came up with this gyroid-based structure, which is a combination of sinuses and cosinuses if you look at how to generate the shape. But what is more important for the cells, it presents a geometry that has very regular curvature throughout the entire scaffold. So this was implanted with colleagues in Liege from the dentistry in a calvarial bone model under a cup where we looked at bone regeneration inside. As we would expect, 4 weeks after implantation, very little new bone was formed. But then after another 4 weeks, we got a marked increase, especially in the case of the gyroid, and not just quantitatively, but also if you looked at the location inside the scaffold, we saw bone growing up much higher into the scaffold, whereas with the controls, it was always close to the calvarium. So here we used principles that were observed in biology, we translated them in mathematics. We used the mathematics to make a proposal for a geometry, which then was tested again. This is now used in a personalized implant fashion, and also large animal experiments are ongoing to compare with what is already available to clinicians at this moment, where we implanted them the bioptos, so that is the gyroid shape, compared to a commercially available shape where we both had at 8 and 16 weeks a higher amount of bone in the regeneration zone. But you can also appreciate that there's still a lot of optimization that needs to be done because there's way too much black to be really a good implant for when we want to put dental implants in there afterwards. And then a final series of examples are about models that try to bring together different things. I already briefly mentioned the one on osteoarthritis. I want to go into a little bit more detail on the one on bone regeneration. I did my PhD on models of fracture healing, but most of the other models that exist on fracture healing, it only started at the regeneration phase. So it completely skipped over the whole inflammatory part. And this is now something that is receiving much more attention. So we developed a model to look at that. You see here the geometry that we use, so you will see this shape here in white as a result, or as a way to show the results. And what we did is we started very simple. Again, a lot of simplification has been made, but starting to implement some principles. And we focused first on the macrophages, how you go from the naive to the pro-inflammatory to the pro-regenerative ones, and the cytokines that they produce, that they are influenced by, and how they interact with one another. All of this information that is in these arrows is coming from the literature, from in vitro and in vivo studies, and was translated into mathematics, and values were available in the literature. We put this all together in an agent-based model, which means that you simulate every single cell as an agent that has to live by certain rules, the rules that come from this graph that I showed, and also the ability to move around. This was implemented. What we then did is we took data from experiments looking at immunofluorescence for these different cell types that we had in the model. And then we focused in the first instance mostly on the amount of M0, M1, M2 over time, the very early phases of fracture healing, where we used our own experiments as a way to calibrate using genetic algorithms to make sure that we had values. This is if we use only values from the literature, and we got the quantitative comparison better by using this new values that we got from the genetic algorithm. And then we had a whole different set of experiments from Kate's lab in Berlin to use as a validation to see that these mechanisms seem to make sense. We're now adding mechanical loading to this to then also get a better understanding of how fixation affects this inflammatory phase. We all know that there are influences, but to be able to put this into the model. So we did the biology groundwork already, and we are now working with these macrophages, putting them in loaded microfluidic chips so that we can get, and we know the validation or the in vivo situation, but we also know the in vitro rules on how we can adapt this schematic that I showed you so that we can continue to gain more knowledge on this. So, told you it was a lot in a very short time. Again, I'm happy to talk about this, but I think it kind of gives maybe a little flavor of how diverse digital twins can be used in all kinds of different applications. So, to acquire new insights or bring information together from very different scales and different types of experiments, to perform screening experiments and to do in silico optimization strategies for new therapies. And it's very important, as with digital twins, that there's always this data part and this model part. Good, so now where are we going with this? Digital twins in healthcare, what I'm showing here was used mostly in the lab. But if we look at the broader field, especially applications where they focus on a single organ or a single scale, like a length scale, we have applications that are already in clinics, that are already reimbursed, especially when it comes to clinical surgery or clinical decision support systems, to surgery planning. These are already a clinical reality. And then there are a whole bunch more models that are actually engaging with EMA to get qualification advice to be used in further studies. And there's even a case, but a very specific case, where a model replaced a full clinical trial. This was only for a medical device focusing on modeling the physics and not the biology, of course, which is more complicated. Now, so we're already there, but there's still a lot of reinventing the wheel. And especially when it comes to combining different organ systems to answer more complex questions, to combine different length and time scales, there's still a lot of work that needs to be done. Europe has already invested quite some efforts into this digital twin field, but they're a bit dispersed. So now the Commission decided that they actually want to come up with a platform where all these things can be gathered so that we don't lose the knowledge that we actually have. And they called this the Virtual Human Twin Initiative that they baptized 2 years ago officially in Barcelona. Now, in order to kick this off, they want to have a roadmap done by the community, and that is what the EDIT Coordination and Support Action was about. We were asked to bring together the community to have a discussion on the roadmap. How do we go from here to as far as we can on the virtual human twin in the next decade? And in order to do so, we also developed a proof of concept repository and simulation platform to inform the roadmap. The first thing was getting together the entire ecosystem, and it's been a bit of a crazy past 2.5 years, but it was really, really great to be able to talk to so many different people that in some way had a vested interest in this virtual human twin. And we had people that are that represent the needs, so patients and civil society organizations, healthcare providers. We had people that are the creators of digital twins, both industry and academia. We had the enablers, which are the infrastructure, so the compute, but also the funding and financial actors. And then, of course, you have the managers, which is the governing framework, the ethical, legal, social actors that are there. It's not so straightforward. There are a lot of people that fit in multiple categories, but we brought them all together, and altogether over 800 people contributed to the roadmap that we put together. Now, how did we address the roadmap? We started, as any good therapeutic design project, with a need analysis. So we looked at the need of different people, all the stakeholders that I showed you, but in particular, I want to show you two categories. Let's say the industry. We had a lot of conversations with medical devices, software, pharma, large and small companies that mostly looked at this initiative of the Commission and the Commission's willing or desire to build an infrastructure, a digital infrastructure around this as a way to access resources, data sets, infrastructure, storage, as a way to, as a sandbox or a benchmark for their own development, as a way to be informed about new technologies, about developments on the ethical, legal, social side, and of course as a networking opportunity. Whereas clinicians mostly saw this as a way it could accelerate addressing unmet needs, improving patient outcomes, reducing cost, hopefully in some way, and then enhancing the quality of care. But with a very important remark that this needed to be integrated in full in the clinical workflow for it to become really something that was used, with access being thoroughly considered, and of course the ways of getting the data annotation done with as little as possible impact on their clinical activities. We then looked at the technology of this. So both the technology of building twins, of generating data, but also building an infrastructure. The infrastructure would be something like this. So we would have access as a user to a catalog where you can find models, where you can find data sets, and then you can use or access workflows like protocols in the lab. You can access workflows that will help you run these digital twins and also run standardized tests that will help you to assess the credibility, because that's very important. We did a lot of work with the regulatory agencies on how to do credibility assessment of digital twins. So this platform should be able to help you with that. Now, data, of course, it's not of creating yet another repository, but it would be creating a catalog that has some data that maybe doesn't have a home yet, but also accesses from which you can access or at least know where to find other data sets, including initiatives that are ongoing like the European Health Data Space, but also initiatives that are more focusing on research data and some other bigger infrastructure initiatives from the Commission. Now, the models would then be like space crawlers. They would crawl over this data space and interact with it by using the right metadata. We have metadata standards for the data itself, while we also have metadata standards that we should use to indicate what models, what are they doing, what do they need as input, what do they deliver as output, so that we can talk or that we can use the infrastructure to find the right data set for a specific model or find a model that can be used on a specific data set. So, of course, this all happens in a framework that is the legal situation that we're facing or that we're confronted with in Europe. Standards are there that can be respected. There's already over 149 standards available that have something to do with digital twins, so we don't need more, but we need to have access to them and implement them. We have a very active regulatory science field on digital twins where we are engaging with FDA in the past and more on the European space as well, where we're evolving towards good simulation practice guidelines. We know the legal situation is a bit complex because we have AI Act, MDR, they're all affecting the use and the implementation of modeling and simulation in our practice. And then, of course, there are all the ethical, the legal ethical aspects, but also the broader ethical aspects that I'm not going into detail into. There is also, because this is touching patients, this is touching healthcare providers, the social aspect, where trust is a very important thing, and in order to work on that, we did a lot of communication, we developed a lot of communication material that we all put together in the info kit. And I'm almost done. So we looked at the uptake and the sustainability. There are these market projections on how the numbers for digital twins will continue to go up for whatever it's worth, of course, because who can look into the future, but also activities on how we can provide services based on an infrastructure that the Commission is going to develop. So all of this was put together and we generated 30 recommendations to the European Commission and other stakeholders for implementing this vision of the virtual human twin. Now, we did so much work that we didn't want to put this in a roadmap of 100 pages and toss all of that out, so we made a 300-page document. I admit it, that's not the right way to do a roadmap, but we more see this as a place where you can go and find this information. We did make a policy brief where we tossed out all of the technical information and we kept the why and the how for policymakers. And if you really don't want to read more than 2 pages, I can advise you to go and look at the manifesto that we created. Because the European Commission, being the European Commission, we thought it would be good to have something to remind them of their intent to work on this. So we wrote a 2-pager, and then we asked people to sign it, not just Lies from the University of Liege, but we asked the vice rectors, directors, the CTOs, the CEOs of these organizations to sign this, so that they said, well, this is something indeed that we are interested in, so you should continue doing this. So with all of that, the little tour that I wanted to give is over, and thank you very much for your attention.
Thank you very much. We are a little bit pressed for time. I have one question. So coming back to the first part of your talk. So you had the examples of the personalized implants or, for example, the fracture healing with inflammation. If you develop something, how would you set up sort of a in vivo validation test, maybe for these 2 examples? I could imagine that for the implants, that takes years before you find out whether implants fit better or worse. For the fracture healing, and maybe faster. How do you do this? How do you test that your model helped to find a better solution?
I would say actually it's the reverse of what you just said with the fracture healing, because we're way lower TRL. So we're very much on the basic side still of the research. So just getting started to get the model up to a level where we have confidence in its predictions before we can then go and say maybe different types of stiffnesses of fixators should be used. It's going a bit in this direction, or in the non-union phase, or in the non-unions, that's how we want to use them. But this is still very early days. With the scaffolds, actually, well, we can do the mouse experiments showing that we have an improvement of the amount of bone formation, and then go to the large animal experiments where they did see that there was a better filling of the entire scaffold. What then happens is that, well, there's still too much material, so we're now taking a step back and going to an improvement of the material before we can then take this further. But these gyroid scaffolds are already being used by Serum, a spin-off of the University of Liege, in mostly craniofacial applications where the mechanics is not so important yet.
And have they tested whether there was any sort of better performance?
Yeah, they did see that there was a better ingrowth of the bone inside these scaffolds compared to the typical lattice structures that they have.
Yeah, thank you very much.